All right, good evening, everyone, and welcome to Genomics Lite. This is the first in our human genetic variation series, and we've got Susan Fairley with us this evening. So we'll be doing a sort of introduction to what human genetic variation is and why it matters for health and medicine and biodata. Just before we start, I'll tell you a little bit about how the session's going to run, especially in case you haven't used Zoom webinar before. So you've probably spotted that you can't use your camera or your microphone to talk to us tonight. Instead, we have a different sort of options for you to do that. So if you need to get hold of us at any point, whether you're having tech issues or you have just a question, you can use the chat box, which is sort of at the bottom right of the screen, and you can just say hello, or you can ask us any questions that you might have. If you've got any sort of science questions or questions specifically for Susan about her work, you can use the Q&A box, which should be at the bottom of the screen. And that makes it a little bit easier for us to find your sort of specific science questions. We'll also be using polls throughout the session, so they should jump onto your screen and ask you a quick question. I know that if people are on mobile, sometimes polls are a little bit difficult to find. So if you're having difficulty finding any of the other stuff, the chat box should always be there, and you can always use that to say hello or ask any questions. Lastly, we are using closed captions today. It is the first time we've used them, so apologies if there are any difficulties with using them. If you'd like to use closed captions tonight, there should be at the bottom of your screen a little CC box, and you should be able to turn those on if you'd like them. Again, this is the first time we've used them, so apologies if there are any issues. But again, you can use the chat box to let us know if you're struggling with them. Before I hand over to Susan, I'm going to give you a little introduction to the campus and our work, and then I'll let Susan take over for the rest of the session. So I'm going to share my screen. So as I said, this is the first in our human genetic variation series. So this week we're joined by Susan. Next week we'll be looking at how the human genetic variation can be researched in the lab with a couple of researchers from campus. The week after, we will be joined by a host of different researchers to talk about career pathways in the field. And on the 25th of March, we'll have our last session of the series with Richard Milne, and we'll be looking at what human genetic variation means in terms of ethics and society. We'll add some information in the chat box in a minute about where you can sign up to those after today's session. So we all work at the Wellcome Genome campus, which is based just outside of Cambridge. And there's lots of different science that happens on this campus that I'll tell you a little bit about in a minute. I actually started working here during the pandemic, so I've actually never been to campus. I currently live in East Scotland, so I'm pretty far away from the campus. I'm interested to know where you guys are all from. So if you can find the chat box and if you're happy to tell us, you could pop in the chat box where you're from and we'll see who's the furthest away from campus tonight. I know occasionally we have America, that's quite fun. Oh yeah, we got someone from New York, London, Norwich, not too far. Quite a few from Norwich. Yeah, quite a few from Cambridge. We've got quite a lot of local watchers from Cambridge tonight. Minneapolis, was that? Awesome. We've got a pretty international group tonight, although quite a lot from the east of England. So welcome from near and far. I'm glad you can all make it. On the campus then we have two main research institutes. There's a couple of other ones as well, but the two main ones are the Wellcome Sanger Institute and EMBL-EBI, which stands for the European Biomatics, European Biomolecular Lab Biomatics Institute, and that's where Susan works, and so she can tell you a little bit more about that in a minute. As the name of the campus suggests, we all research things to do with genomics, and so I've got a little poll that I'm going to pop up that's going to ask how much you know about genomics. This isn't a test, this isn't to, this is just to help us know who's in the room, as obviously we can't see you tonight. So I'm going to launch that poll and hopefully that's jumped up onto your screen. So you can just pick an answer. If you can't find the poll, feel free to put an answer in the box about how much you think you know about genomics. All right, we've got quite a mix, which is great. So quite a lot of people have either a limited or fair knowledge, which is great. That's why we host these sessions, to help people learn a little bit more. Awesome. On the campus, then, everything that we do is to do with genomics, but there's sort of 6 main areas of research. We've got human genetics, cancer, cellular genetics, parasites and microbes, the tree of life, which is looking at biodiversity, and biodata. So our talks for today sort of fit within the human genetics and a little bit of biodata as well. And lastly, before I hand over to Susan, I just wanted to point at some useful resources, especially if you're studying for GCSEs or A-levels or anything in that sort of age group. Our Your Genome website has lots of articles, videos, resources to help you learn more about genomics. You can sign up to our education update. This is where we update people as to what we've got going on. So it's where you can find out about new series of Genomics Lite and other activities that we have going. You can connect with us on social media and you can drop us an email as well. Again, I'll put all the links for all this stuff in the chat box in just a second. I think that's all that I wanted to touch on before handing over to Susan. So I'm going to stop sharing my screen. And Susan, if you're happy to take over, I will pass on to you.
That sounds great. Thank you very much indeed for the introduction. And if I can ask people to bear with me for just a second or two, I am going to try and share my screen. I think I have found the right window, and hopefully you are going to be seeing the title slide for my talk, which should be saying Human Variation, the 1000 Genes Project and Beyond.
Yep, that looks great.
That's brilliant. Okay, so as Em said, my name is Susan Fairley. I work at EMBLI, which is the European Molecular Biology Laboratories European Bioinformatics Institute, and we're based just outside of Cambridge. And I work on a project called the International Genome Sample Resource, which was set up to kind of follow on from the 1000 Genomes project. And I'll talk a little bit about that as we go through the talk today. But what I'm hoping to talk to you about just now is a little bit about human variation in general, then a little bit specifically about the 1000 Genomes project, which was a project that ran from about kind of 2008 to roughly 2015 to explore human genetic variation. And then we're going to look at where things have gone on since then in the remaining time. So if I move through to my first slide. So one of the questions when we're talking about human genetic variation is, what is a genome? And Em has mentioned that we've got some polls lined up, and I believe this is the first one. So I'm going to ask Em if she will work her magic and share the poll with you. And if you'd like to tell us what you think a genome might be.
So again, a nice poll should have popped up on your screen, but if you can't see that, then you can just give us an answer in the chat box as well. Excellent. We started with a not too challenging one, but it looks like most people have got it right. Only one person has fallen for my type of gnome answer. So it looks like pretty much as everyone has got the right answer, which is all the genetic material found in an organism.
Thank you. And that's absolutely correct. So what a genome is it's the complete complement of genetic material. So we've got the chromosomes here and also the mitochondrion is part of that. But it's basically it's the full set of DNA, the inheritable material that's going to get passed on from one organism to another. And as I say, we've got a picture here looking at some human chromosomes. We've got them listed 1 through to 22 plus copies of the X and Y chromosomes. But that then brings us to another question. If this is covering the full set of genetic material that any one individual has, what then is genetic variation? And so I think Em has also mentioned that in addition to doing polls, we also have a chat box. So it'd be really interesting if some of you might like to take the opportunity to use the chat box and maybe let us know what is it? What do you think we mean when we're talking about genetic variation? What is genetic variation?
If you've got any ideas or maybe just whatever comes to mind, if you hear the word genetic variation, which I think we hear more in the news than maybe we ever have before. So we've got a few suggestions. We've got gene mutations, which is a good one. Epigenetic changes, that's a pretty specific one. Oh, there's lots of answers coming in. Diversity is coming in, how individuals differ in a species. That's a really good one. Yeah, mutations and alterations, variations, alleles. Yeah, lots of excellent keywords being thrown around in the chat.
Okay, well, that's really great. And I think of those suggestions and answers that Em shared, you guys have hit the nail on the head. So what it is it's alterations, it's mutations, which may be in a gene or maybe elsewhere. And if we go on to look at this slide, this is a kind of cartoon of a very simple type of variation, where we can see in the top part of this, we've got a sequence of bases, AATCGC, GTAC. But we can then see that in another copy of the same region of DNA, we've got a switch has happened. And we've got that second C, the one that's highlighted in red in the top sequence, has switched over to a T in the lower one of these two. And then this would be a single base change, so a point mutation, or if it occurs in a certain level in the population, that's going to be a single nucleotide variation. That's one type of variation. However, there's actually lots of different ways and categories of variation. And in some senses, these categories are a little bit artificial, but they're useful for us in terms of how we think about variation. So if we look at the top left of this slide, we can see that we've got, as I just said, we've got SNPs or SNVs, as they're sometimes also called single nucleotide polymorphisms or single nucleotide variants, where we've got a single base change. And in this case, we've got a G that has switched over to an A in the lower one of these two sequences. Below that, we're looking at multinucleotide polymorphism. So there's nothing to say that a genetic variation is simply a single base change. It could be that a number of bases have changed. And as we look at the second example in the panel on the left, we can see that there's a series of 3 bases that have changed. So a TAT has become a GCG. And so this is a change which is affecting multiple nucleotides. It's also the case that it's not the case that these two categories that we've looked at so far are substitutions where a base is simply changed for another one that's been dropped in. But it's also the case that sometimes these things can either be dependent on which of the two sequences you're looking at, either an insertion or a deletion of sequence. And we've got an example of that at the bottom of this one, where we've got what would be a deletion in the top sequence or an insertion in the bottom sequence of the bases G and A. But these are all looking at variations in the genetic sequence that are relatively short. But we also get much larger variations that happen over greater lengths. And it's a kind of arbitrary cutoff, but we have this cutoff of 50 base pairs in length. And we say that if variations are greater than 50 base pairs in length, then it's considered to be a structural variation. And these can, we've got an example here where we have 4 blocks, which we are imagining are greater than 50 base pairs in length, A, B, C, and D. And it could be that one of these blocks gets deleted. So as in the second row of this example, block C has gone. Or it could be that there are duplication events, and you may sometimes get many copies of the same region of DNA repeated multiple times in a given chromosome. So we've got single nucleotide polymorphisms, multiple nucleotide polymorphisms, insertions and deletions, and these are all happening at a shorter range. And then we've got these larger structural variants. But then on top of that, we've also got what's the number of different versions of this one given variant that we can get, because if you look at a kind of simple example in the bottom right hand panel here, you've got how many different alleles or how many different versions of this particular variant exist. And if we look at if we look simply at individual 1 in the example, we can see that for their 2 copies, we've got G and A, so that individual would be biallelic if we were just considering that individual. However, if we were then to look at that variant across multiple individuals and we take an individual 2, then we can see that actually we've got 3 different alleles occurring at that location. We've got G, we've got A, but from individual 2, we've also got T. And so if we see just 2 versions of a given variation, we call it biallelic. If it becomes more than that, then it's termed as being multiallelic. Okay, so that's talked a little bit about a kind of the molecular level, what variation looks like. But what does it look like when we move on from that? So these are 2 of my colleagues. On the left, we have Laura, and on the right, we have Ewan. And on this slide, we've got a list of about 3 different traits, and we're saying what characteristics Ewan and Laura have for each of these. And the first one is eye color, and we've got height, and then we've got accent. And what we're interested in thinking about here is what's the genetic basis, or what might the genetic basis be for these? And I think we have a series of poll questions for each of these. And I think first up is going to be eye color. So our question is basically going to be, do you think that it is determined by genetics, what color somebody's eyes are? Is it down to environment, or is it down to a combination of the two? And so I'll let M share the poll question.
So this poll is actually a combi poll. It's going to be 3 questions in one, which is exciting. So the first question will ask what influences eye color, the second, what influences height, and the third, accent. So the answers are either genes only, environment only, a mixture of both, or neither. So I'm going to launch that poll. You can have a go at answering all 3, and we'll see what everyone thinks. Awesome. We've got a mix of answers coming in. So it's looking like for our first question about eye color, most people think it's only genes, and a few people think that it's genes and the environment. So maybe you want to talk about that one first, and then I'll tell you how the next bit of the poll is looking.
Okay, sure thing. So I'm going to agree with you on that one. So yeah, it's genetics that's responsible for eye color. I don't know. Do you want to go on to the next step of the poll?
Yeah. So height is almost the exact opposite. Most people think it is both genes and the environment, and some people think it's just genes only. So almost the direct flip from the last one.
So I'm going to agree with everybody once again. Height is indeed a mix of genes and environment. And if we think about it, height's in some sense is one of the kind of classic examples of one of these traits, which it's when it's down to a mix of genes and environment. And one of the things that and people often kind of talk about when they're thinking about this is that there are a number of different genes that influence how tall you are. So you have a set number of genes, and you might have, the tall or the short version of each of these different genes. But let's imagine, just purely for the sake of argument, that there were maybe, say, 5 genes that were going to influence height, and you could have either short or tall versions of these 5 genes. So you're then going to have a fixed number of combinations of genes that would be dictating height. But what we don't see is people who just fit at discrete intervals in terms of height. Instead, if you look at the population, you see this smooth gradation of height right across, and it's because it is a mix of a number of different genes and also environmental factors like how good your nutrition was when you were growing up and things like this. And between the two, the interaction of the two, it means that we end up with this kind of smooth gradient across that trait. I'm sorry, the results for the final poll, please.
Yeah, no, I was going to say the way I've always thought about it is you could have all the tall genes in the world, but if you ate nothing and did no exercise, you're not exactly going to be giving your body the opportunity to get really tall. So for our third one, for the accent, we've got quite a mix. So nobody thinks it's only genes. Most people think it's the environment only. A few people think it's both genes and the environment, and some people think it's neither, which is interesting.
Okay, so for this one, I'm going to go with environment. Because I think when we pick up accents, we're picking it up from what we're hearing round about us. So all of us are learning to speak, and some of the biology that's involved in our learning and brain development and stuff, sure, that's going to come from genes. But in terms of our accent, and specifically whether we sound like we're from London or Aberdeen or Manchester or Newcastle, that's dependent on the voices that we're hearing round about us as we grow up, and it's also something that can change if we move around it during our life, and we can pick up different accents. So moving on, this is kind of basically what we've got for these 3 different things. So we've got Mendelian down for eye color with a little asterisk, because it's not really, but it's pretty close to that in the way in which it behaves. So it's pretty much dictated by genes, and it's a relatively straightforward pattern of inheritance. For height, it's much more complicated. It's a complex trait, and it's a mix of multiple genes and environment. So then this would also apply to other traits. For example, blood pressure is a good example of one that's a complex trait. And then for accent, that's going to be something that's pretty much dictated by where you are and what's around about you, rather than coming from your genetics. So This is looking at what we see as Laura and Ewan, and for these three different traits in terms of what we see when we look at them or interact with them and talk to them. But what about at a genetic level? So it so happens that Laura and Ewan have both had genetic testing done, and we can see that this is looking at the results for eye color, and we can see that, yeah, actually what we see with Laura and Ewan, it matches up what we would expect to based on their genetics. So Laura has a thing called the genotype, AG. So she has one copy of A, one copy of G for this. And yet Laura does indeed have brown eyes, and we can see that Ewan is GG, and indeed, as expected, has blue eyes. But what do we mean when we're talking about a genotype? So genotypes refer to which versions of the alleles that people have. For the bulk of our chromosomes, we have 2 copies of them. And that means that we have 2 copies of any given gene typically. And if we have 2 copies that are the same, it's described as being homozygous. And if we have 2 different alleles, then it would be heterozygous. And the example that we've got here, and we've got it illustrated as this individual is for our locus of interest is AG. So this would be an example of heterozygosity. As we continue looking at kind of different types of variants and exploring them, there's also another kind of distinction between variation types. And this is a distinction between germline and somatic variation. So germline variation is basically what is passed down through the generations. It's what you've inherited from your parents. And we have a photo here of Laura on her wedding day, surrounded by her family. We've got her sisters and other family there. So germline variation is the stuff that's getting passed down through the family. Somatic variation is different. Somatic variation is something that has occurred spontaneously. So you're going to have started off with a cell somewhere in your body. Let's say it's maybe a skin cell on my hand. And at some point, as the cell is going through its cycles and continuing to divide and so on, as it works through the replication process, mutations can occur. And when mutations occur like that, it's referred to as somatic variation. And it can be particularly found in some tissues, and is also one of the things that sometimes, if there's a sufficient accumulation of these, and specific mutations in particular, these are the types of mutations that are more commonly associated with cancers. Okay, so that's talked a little bit about different types of variation. If we're looking back to kind of more to the kind of germline variation, what's the origin of variations? How have they accumulated in the population? So we have this kind of a little diagram to kind of work through. And if you look at the kind of bottom right of the screen, you can see that we've got a small segment of sequence, which we're going to use as an example here. And we can imagine an initial population, which we're seeing in the map on the left-hand side. They're starting off in Northeast Africa. And we can see that these groups of people have moved, and populations have moved out. And we can see that we've got a couple of different variations that have come in here now. So we now have a T has changed on the population that's in the left-hand branch, and we've got a G that has been introduced on the right-hand branch. And so there's been an original population. This has then spread out to create an additional 2 populations, and within those populations, certain variations have emerged. This process can then repeat, and we can see further additional variations accumulating. So if you follow the right-handmost branch here, we can see that original G, there's now a T in there as well, and so on. What we can also see is that sometimes we're going to see different populations coming together, so that variations that were seen in 2 separate populations are then going to be merged and mixed as a new population has formed that is combining different populations. So it's not simply a branching process which keeps on getting narrower and narrower. It's actually a much more fluid arrangement where it mixes and merges and splits up and comes back together again. We can see this continue until eventually we can see that we've reached a stage where we've gone from our original sequence at the bottom of this slide up to about 5 different variations of this sequence, each of which has got different combinations of alleles at different locations as we've moved through this time span, looking at over the course of these thousands of years. Okay, So that's going to wrap up what I want to say really about the introductory part of this talk, exploring genetic variation. I'm then going to move on to look a little bit more about kind of recent or more recent projects, because I'm looking at the data on this, since this is a quote from 2000, so it's now 21 years ago. But 2000 really was around a fairly major kind of turning point in genomics, because it was going to be about 6 or 7 months later that just at the beginning of 2001, that the first version of the human reference genome was published. And as they were approaching the end of the Human Genome Project, they're starting to look ahead to the future, and they're looking to say, you know, okay, we've done this, we've now got a reference human genome, singular. What comes next? What you know, look into the future. And this guy, Sidney Brenner, included this in a piece in Nature Biotechnology. He said, after the first complete sequence of a human genome is obtained, the next challenge will be to discover and understand the function and variation of genes, and ultimately to understand how such qualities affect health and disease. A key to this undertaking will be the availability of methods for efficient and accurate identification of genetic variation. So at this point in time in history, what's happened is that they've managed to put together a kind of a map of what we think a reference human genome is. So it's not just one person, and it's got blocks from one person and blocks from another person, but it only represents a single person's worth of genome. And that's incredibly useful for researchers in terms of understanding things like this gene is near this gene, or this gene may be controlled by this section of sequence or whatever. But what Sidney Brenner here is saying that actually, if we want to kind of move on through and understand what's going on in terms of function and variation, and the impact that genetic variation has on health, in order to do that, we need to be able to look at genetic variation more widely. And if we're going to do that, we need the tools and the technology to be able to do that.
Susan, we've had a couple of questions that I think might sort of fit in just around what you're saying about the Human Genome Project. Sort of who, and I wonder if you know the sort of answer to this, whose genome was sequenced in the original Human Genome Project, and how did they choose that?
And that's an even better question, how did they choose? So there's actually a lot of different stuff around about this. I don't have the specific answers at my fingertips, but I have a good idea of where you would go to look for it. So the people who are the kind of current custodians of the human reference genome is a group called the Genome Reference Consortium. And I would suggest that taking a look at their website, they would probably have some information there.
We've actually had a previous speaker talk about this, so I think me and Fran know a little bit about this, only a little. I believe, Fran, correct me if I'm wrong, it was anonymous, and they asked for anonymous donations from about sort of 6 or 8 people, so it wasn't just one person's genome, it was a mix to start with, and it was anonymous, but I think most of them were European and American, which is, I think, something that you're going to touch on in a little bit, maybe the limitations of doing that.
I think I have once been in a talk where I've seen people share the newspaper advert, because they placed an advert for donors. Although there's, as I said already, it's not just one person's genome, but there's a few different ones in there. But yeah, so one of the things about moving from having a single reference genome to exploring genetic variation is that you need this change in the tools and technology. Now, the Human Genome Project was a big project. It ran over a number of years, and it cost really quite a substantial amount of money. So this was not a trivial undertaking. And I think we have a poll question now where we're going to ask you if you would like to basically pick a number from our list and tell us how much did you think it cost for the Human Genome Project. What was the price tag for producing the reference human genome?
So I popped that up on the screen. You can always put another number in the chat box if you can't see the poll this time. Oh, we've got an interesting mix. So no one thinks it was 1 million. That seems to be too cheap by the audience's guess, but there's a good split amongst the other ones. So about 40% of people think it was 500 million or 500 million. 48% of people think it was 3 billion, and a few people have gone for 600 billion. So most people think it's somewhere in the couple of billions mark.
Okay, and that's a pretty good answer. So again, for emphasis, this is back in 2001. So what you can buy with your money has changed in that time. But in 2001, getting to what was judged to be a working draft. So it's not completely finished. There's still gaps. There's still spaces in it, and it's not perfect. But something that you could start to work with. That cost $3 billion, and it took 10 years to get there. Now, we have just spoken about the idea of needing the tools and technology to be able to look at more genomes if you want to explore variation. And you can say that, well, if it costs $3 billion and takes 10 years, that's a lot of resources to start and look at multiple genomes. However, technology's changed and moved on, and we had what in the world of bioinformatics still even now, quite a long time later, is referred to as next generation sequencing happen. And this started to really become available around about 2008, which was the point at which the 1,000 Genomes project started. And this was really pivotal to the 1,000 Genomes project being able to start. And at this point, with the arrival of the new technologies, things changed drastically. And suddenly the major genome centers can sequence the same number of base pairs that they sequenced in the Human Genome Project, not over the course of 10 years, but they can do it in 4 days. And this just was really a complete game changer. And the data is of a very different type. Reads are much, much shorter. The way in which you would work with it is really different. You can't do the same things with it that you could do with the Sanger sequencing data that the Human Genome Project developed. But suddenly, Sidney Brenner's idea of, hey, maybe if we have the right technology, we can start to look at more genomes and more variation. Suddenly, that's not looking so improbable. And what happened after 2008, though, was that things didn't just stand still then. The technologies continued to change. And they changed. They didn't just continue to change, they changed rapidly. So by 2009, just 1 year later, that volume of data has gone from every 4 days to every 4 hours. And you can produce that volume of data for only $25,000, not $3 billion, but 25,000. And again, it continues. By 2010, again, just 1 year later, you can now produce this in 14 minutes and it costs $5,000. And by the time we get to 2015, the cost is down to around about $2,500. So in this relatively short period of time when these technologies arrived, things changed drastically in the world of genomics in terms of how much data and the cost of data that could be produced. So suddenly, these tools that would be needed to start to look at lots of genomes, suddenly this is now possible. This is looking like something you could do. And again, just for emphasis, this slide is now a few years old. But this is kind of to illustrate the change in the cost of sequencing a human genome in this period of time. So on the left, we have what I am told is a photograph that is representing the most expensive house in London. Apparently, you're not allowed to take a photograph of it. It's that expensive. But in 2003, that's the sort of money that you were talking about. By the time you've moved to 2016, you're looking at about the cost of an Arsenal season ticket. So it's not cheap. It's not your loose change from down the back of the sofa, but it's a very big difference from where it was back in 2003. And as I said, this is really then what drives and makes possible things like the 1,000 Genomes Project, which set out to actually look more broadly at human genetic variation. So the 1,000 Genomes Project's aim was to catalogue common human genetic variation, the types of variation that people who are wandering around in the street feeling perfectly healthy and fine, not necessarily with any particular illness or genetic condition, but just what's the variation that exists in the wider world and in people in general and at large. And the project ran from 2008 through to 2015. And it set about to develop the tools that would be needed. These data types are completely new. This is never something that anybody has been able to do or attempt before. And even really kind of mundane things like working out what the file formats are going to be. How are they going to store this data? How are they going to work with it? What's it going to look like? And they worked with this using a set of samples from 26 populations across 5 continental groups, and looking specifically at data from 2,504 unrelated individuals. And the idea with this was that they were going to be able to use this to produce reference data that could be used by biomedical researchers. The cell lines that they produced, as I've said, are from across 5 different continental groups. They're held at a place called the Coriell Institute. And one of the things about the cell lines is that it means that it's possible for researchers to come back and revisit these samples. So either as technologies change and develop, in terms of sequencing technologies, you can come back and produce data on the same samples and see, how does this differ? Is it making a difference to the quality of the data that you're getting back? And it also means that if somebody sees something interesting or an interesting variation or something in one of these samples, they can go to Coriell and they can get access to these cell lines and do further study. One of the other things which is unusual about the 1000 Genomes Project is the samples that were in the 1000 Genomes Project have an extremely open consent. So there's no phenotype information is collected. We don't know anything about these people. Were they short? Were they tall? Did they have high blood pressure? Also, they collected more samples than they used, so it's not possible to necessarily identify anybody or know, even if you found out that somebody had contributed a sample to the project, you wouldn't then automatically know that they were part of that set because they may or may not have been sequenced. The participants were over 18, and all of the data and cell lines can be shared freely without restrictions. So this means that people in academia can work with them. If you want to say you were doing a training course or something, or you were learning about how to look at and discover genetic variation, you could work with this data set. It could be shared with you freely. And if you were in industry or whatever, there's no restrictions on the use of this data. So it's made freely accessible to anybody for any purpose. So what was the basic strategy of this 1000 Genomes Project for looking at variation? So basically it boils down to this, which was that they were going to, once they got their samples, they were going to sequence the DNA and collect the sequence reads. And reads is just a term for a string of DNA, a string of bases that have come off the sequencing machine. So that's what we're talking about when we talk about reads. And then what they were going to do was they were going to align these reads back to the single reference genome that existed at the time. And then based on looking at and comparing these reads to the reference genome, that's how they were going to detect a variance. We have a slide here which gives some kind of an indication of what this would look like. So this is looking at some really old data. It's very short. And what we have here in this slide is across the top of the slide, we've got a string of characters and a couple of red asterisks. And this is the reference genome sequence. And then below that, we've got these set, in some cases, it's just like a little row of dots. And where it's contiguous, this is a read, and where it's just dots, it means it's the same base as is listed in the reference sequence. We can see that I think the third or fourth sample down, that we have one where there's an A has crept in instead of the C that's in the reference sequence. Moving a few samples further through, we can see that there's a G has been a substitution. And if we look over in the middle of the slide where we've got the set of asterisks, we've got what appears to be an insertion, and it looks like in about 3 of our samples, the bases A and G have been inserted in the reads that have aligned there. So this is a fairly basic representation of these alignments. There's other slightly more kind of sophisticated viewers. So this is looking at a view of aligned sequence reads or aligned back to a reference genome using a piece of software called IGV. And we can see here that in this case, yeah, we've got a bunch of reads that have been aligned to the reference genome. And if we look here, we can see that we've got what looks like a different allele, in that we can see that there's a few of these cases where we've got a C instead, but it's not all of the cases. It's not for all of the reads that we've got there. So this would then kind of start to raise the question, if you're looking at data like this and you see this, well, is that a variation or is it not? We can see that some of the bases have changed, but how do we decide, and how do we, where do we draw the line? What's the cutoff? And there's a number of different factors that might come in at this point. And I think I'm correct in saying at this point, I think we have another poll question now.
We do. And there's actually just a couple of questions about your previous slide. Maybe if we could jump back for just a second.
Sure thing.
On the black slide with the... Yeah, that one, that's perfect. There was a question about what the asterisk meant and then what the double N meant as well. I'm not sure why the double N, what do the two N's mean? Yeah, the double
Okay.
N on the read halfway down and what the asterisk meant as well.
Great questions. So the asterisk in this case means that there's an insertion. So it means that in the reference sequence at the top, there weren't any bases at this point, but we can see that in some of the sequences, so the three in the middle where we've got AG, they seem to have bases that when these reads have been aligned back to the genome, the three reads that have got AG in them, there hasn't been an A and a G to match them to. So instead, the alignment software has decided, okay, I can't match these here. So instead, this should be an insertion. And so when we look at this, which is essentially almost kind of like a multiple sequence alignment, what we're seeing is these two bases are appearing as an insertion in the reference sequence and also in the other reads where there is where these two bases are absent and where there's a better alignment in another way. So the asterisks are indicating an insertion. The ends are what is called an ambiguity code. And there's a number of different ones depending on what combinations of bases you might think it is. But it's sadly the fact that when sequence data is generated, it's not always the case that it's actually a clear-cut answer as to what the base is. So sometimes the data that comes off of the sequencing machine will indicate that, yeah, there was a base here, but actually we're not quite sure which one it was. And if it's the case that there's one where we don't know what it is, typically the most common thing that is done is to put in N. So N is actually basically, you could almost argue that it should be a question mark, because it's like, we know there's something here, but we're not sure which base it is. So it's what's called an ambiguity code.
This looks a bit more professional than a question mark.
This is true. This is true. Okay, so moving through again to the IGV slide. So it's saying, one of the things we have when we're looking at this thing, and actually the ends are actually a great example of that, because there are times when we don't actually know what it is, because sometimes the quality of the data can be variable. So when we do get to the stage where we've got these alignments, we're now at the stage where we can see, okay, yeah, this differs from the reference genome. There's another base there, or maybe it's not clear what the base is at a given point. But it leads us through to the question of what might the difficulties be in terms of calling bases, and what are the factors that might influence how difficult it would be to call a variant at a given location in the genome. So I'm going to hand over to you for the poll.
Awesome. So we've got another poll that's going to jump on our screen in a second that is going to ask what factors might affect whether we call a variant, as Susan was just talking about. So have a go at answering that. A little bit more tricky than some of the previous polls, but from far. Wonderful. Looks like everyone's doing pretty well. So most people have gone for all of the above. And for anyone who can't see the polls, the options were the depth of the read coverage, the quality of the alignment, how reliable the base call was when read, or all of the above. So most people, nearly 90%, have gone for all of the above.
And you're absolutely right in doing that, because all of them can have an impact. So depth of coverage, when we talk about depth of coverage, and I should have possibly mentioned this before asking you to answer the question, depth of coverage is the number of reads that have aligned at any given point. So what's the depth of the number of reads at the given location? And clearly the more data you have at a given point, often the more clear-cut the answer will be. If you've only got maybe a couple of reads and one of them says one thing and the other says the other, that's kind of difficult to figure out. Whereas if you've got a great big stack of reads at a given location, that makes it much easier. Again, also the quality of the alignment, that matters. If it's an area where the aligner has really kind of struggled, then that's a bit more ambiguous, and it's going to be harder to figure out. And also things like the quality of the base, how confident was the sequencing machine when it produced that base? Is it one that we're absolutely certain is right? In which case we're going to say, well, yeah, if we have a really high confidence in this base call and it's a good alignment and we've got lots of data there, then that looks like it's a pretty good basis for saying that, yeah, there's a variation here. Whereas if it was maybe a lower quality call, this sequencing machine wasn't so confident when it made the call about the base, then when we come to align it, maybe also if there's not an awful lot of other evidence there, maybe if also the aligner has struggled a bit there, overall you're going to have less confidence in that call. Okay, so this is basically just kind of summarizing that again. The question that you are then faced after you've looked at these alignments is it really a variation? So are there enough reads there to be sure? Do the reads agree with each other? How good was the base call made by the sequencing machine in their first place? And then finally, how good is the alignment? So these would be some of the factors that you would be thinking about when you're trying to detect variation. So this was some of the things that the 1000 Genomes project had to look at and had to assess and when they were developing their methodologies. But when they applied the methodologies that they did come up with using the strategy that we just talked about taking these short reads, lining them back to the reference genome and making variation calls, this is what they came up with at the end. So this is a figure that is taken from one of the final papers from the 1000 Genomes project published in 2015. And what this is showing is it's showing a pie chart for each of the 26 populations. And the pie charts are split into 4 categories. And in dark colors, we've got the variants that are found in that population and that are only found in that population. They're not found anywhere else in any of the other populations. Then in the paler color, we've got variants that are found within that continental grouping and they don't appear on other continents. And then after that, we move into the pale gray, which is variants that are shared across continents, so they're appearing in more than one continent. And then we move to dark gray, where we've got variants that are they're common everywhere. They exist on all of the continental groupings that the project has looked at. So this is one way of looking at the variants that were detected by the 1000 Genomes project. Another view is to look at variants per genome by population. And if we look at this slide, it's the same color coding. So we've got European genomes in the blues. We have got East Asian genomes in greens, South Asian genomes in purples, moving to the Americas in red, and then genomes of African ancestry in the yellows and oranges at the right-hand side of the screen. I think I am correct in saying once again that we have another poll question for people at this point.
Yes, we've got a poll relating to why are genomes in people with African ancestry higher than other groups. I want to launch that poll. So we'll see what you all think. It's a fairly wordy one, so it might take a little minute for people to have a read. Awesome. Looks like we're getting quite a good number of answers in. So a little bit of a split. We've got some people going for the answer that Africa is a large and diverse geographic area. Some going for humans leaving Africa were subject to strong selective forces, and some going for humans leaving Africa created a bottleneck. And then most people, sort of 60%, are going for all of the above.
Okay, so it's an interesting question. The reason that or the explanation that usually is gone for this is usually it's basically the third option here. So this kind of comes back to this idea of out of Africa, and the idea that humans originally as a species existed in Africa, and that meant that there was a period of time when there is a population of humans in Africa and genetic diversity is accumulating. And this was a kind of state of affairs that went on for a kind of fairly prolonged period of time. But then eventually people left Africa. But it wasn't everybody who left Africa. It was a small number of the people who had been in Africa that left Africa. And therefore, a relatively small pocket of the variation that was present in Africa that then carried on out into the wider world. So usually when we're thinking about what we see in this slide, the explanation that the change in the levels of variation and the massively reduced variation that you see in the rest of the world is because basically there's this kind of pinch point where it was a very small number of people. So there's effectively this kind of genetic bottleneck when people were moving out of Africa. So this is looking again, as I've said, at the numbers of variation in each genome across the continental groups. Another way of looking at the variations that was found by the 1000 Genomes project is looking at the kind of variations by category. And in this slide, what we've got is variants per genome by population and by variant type. And again, the colouring is the same. So yellows and oranges are still Africa, blue is still Europe, green is still East Asian, purple South Asian, and red is the Americas. And if we look at these categories running from left to right, we've got different kind of, if you like, biological categories of variation. So we've got all variation in the leftmost panel. We've got synonymous variation where a base has changed, but we wouldn't expect it to have any effect because it's a synonymous change. We've got non-synonymous changes where we think that this would then continue to have an effect, loss of function and so on. But then as we move over to the right-hand side of this slide, in particular, if we look at ClinVar and GWAS, so ClinVar is a database that records clinical variation data. So it's variants where we have clinical knowledge about them. And GWAS, which is referring to genome-wide association studies. So these are studies that are done typically at a population scale to look for associations between variants and some given traits. So for example, maybe high blood pressure or something else. When we get to these two kind of, if you like, medicalized ways of looking at things, actually the proportion of variants that we're seeing represented there, that's really changed. So it's in the other categories, African variation is dominating. But when we move along, European variation is really prominent, and actually a bit from the Americas, maybe a little from Asia as well. Does anybody want to maybe make a suggestion as to why that might be the case? And you could use the chat box for this. I don't think we have a poll for this one, but you could use the chat box if you have any ideas as to why it might be that when we look at variation by category, we see African variation dominant, as we saw when we were looking at the genomes previously. But when we look at medical variation or variation where we have additional medical knowledge, it's primarily the European variation that is dominant. Any suggestions for why that might be?
Got a couple answers coming in. One that says we have access to more clinical data here. Another that says, is it anything to do with the reference genome being mainly from European people? We might not have as much research on African genomes. An interesting idea that there was an early mutation in European populations. And then another answer around less data in Africa and more research in Europe. So quite a lot of interesting answers.
Yeah, and those are really interesting responses. And yeah, I think it sounds like at least a few of you have basically hit the nail on the head, because what we're seeing here is not so much a property of the variation. It's not that there is a lack of, say, variants that might be associated with disease in Africa, but what we're probably seeing here really is database bias. So when we look at the GWAS databases and entries in databases like ClinVar, these are very heavily based off of data from European populations. So it's not the case that there might not be medically relevant variation in African genomes. It's the case that they're not represented in those databases. So when we're looking at this type of categorization, this probably is just reflecting the essentially the biases in those data sets. Okay. So we've talked now a little bit about general ideas about what genetic variation is, the approach that the 1000 Genomes Project took to cataloguing variation in terms of their methodology, and what the variation was that they found. But how could you use that data? So there's actually a lot of different potential uses for this data. I've only listed a few of them, but there are a number of others. So one of the ones that you can do is you can essentially, if you've got a good catalogue of variation, then actually what you can do is you can use it to kind of make a guess where there's missing information in association studies. So one of the things is that, just mentioned, we talked briefly in the previous slide about these ideas of genome-wide association studies. To do these things well and to get enough power to pick up effects in them, you need a lot of data. So if you want to try and figure out if a gene is associated with some given condition, where maybe the gene is only actually one of many factors that are contributing to it, in order to have sufficient power to pick that up, you need an awful lot of people involved in that study. And that becomes really quite expensive to generate data. So then what you might find is that if you're going for a kind of relatively minimal data generation strategy for, say, your GWAS study, then what you actually may then want to do is find that you're not going to have the information for each individual at each of the locations that you're interested in. And at that point, it's then useful to have a reference framework that you can basically use to help you kind of fill in the blanks. And if we look at the example that we've got at the bottom here, we've got 4 samples listed, and we've got half a dozen different locations in genome for half a dozen different loci, and we've listed the genotypes for these individuals at each of these loci. Now, bearing in mind that we know that genetic material tends to be inherited in blocks, so if you've got a block of genetic material, all of the variants on that are going to get inherited together. We can then look at this example and we can say, okay, well, we can see that all of these samples at the first location here have GG. And then we can see that on subsequent locations, again, they've got the same genotypes with maybe I think one exception. But we can see that for the second sample, sample 2, we weren't able to directly discover what genotype it had. But we can see that all of the other samples have the same genotype at either side of this in these locations. And so actually what we could probably then do is say, well, we know that this genetic material is always inherited together as a block. So even though we don't have the information for the second sample at the second loci, we can actually probably make a pretty good guess that if it's the same as all of the others for this block, that probably it's going to be the same. And that's really important for doing some of these kind of really large scale studies like GEO. So you can use it for the term for this is called imputation. But it's important for that. Another thing that is hugely important is for work in identifying the non-pathogenic variants when you're looking at gene hunting work. So if somebody has got what's believed may be a genetic condition, if you sequence their genome and compare it back to the reference genome, you're going to find lots and lots of variants. And what you want to be able to do is then say, okay, I want to be able to filter out the variants that I know don't cause disease necessarily, or that don't cause an obvious disease. And at that point, that's when having a catalogue of variation from a wider healthy population becomes really useful, because then what you can do is you can compare the variants that you found in the individual you're interested in with this wider catalogue of variation, and you can use this to filter out. And another one is the topic that we touched on when talking about imputation, is this business of identifying blocks of data that are blocks of sequence that are inherited together, which is called haplotype structure. Okay, so that's some of the uses of the 1,000 genomes data. The other thing that 1,000 genomes did, though, as I said already, is it pioneered the development of a lot of these techniques and methodologies. And it's now been possible for those to go on and be applied across a much wider range of much larger projects. And so there's a lot of kind of new medical and both medical and research projects. There's Genomics England, the UK 10K, Precision Medicine Initiative in the United States, the International Cancer Genome Consortium, and also Genome 10K. And all of these have been able to take the changes in sequencing technology and apply them to studies across a wide range of different areas, frequently associated with health. But this also means that we're seeing more and more data being generated. And that was one selection of projects, but there's also more data being generated on the 1,000 genome samples as people develop new technologies. And that has been added into alongside the 1,000 genomes data and the project that I work for, the International Genome Sample Resource. And the net result of all of this is that over time, more and more sequence data is becoming available. So it's possible to kind of build up this library of resources of sequence data that can be used both, some of it in clinical applications, but also by the research community worldwide. And that is one of the functions that the EBI seeks to perform. So one of the things that EBI does is it hosts a lot of archives of data, which provide access to scientists to this data. And just to finish up with a little bit more information about EBI, it's an international non-profit research institute that's part of the European Molecular Biology Laboratory. It aims to be a hub for biological data and research. A lot of the resources are public, and you can take a look at the things there. It's home to the archives, and it has a good number of staff and an awful lot of data. So just to kind of summarize and to finish up, we've talked about what a genome is and what genetic variation is. We've talked a little bit about how changes in sequencing technology have changed what's possible in genomics and have made looking at many genomes possible. We've looked a little bit at the 1,000 Genomes project about the strategy that they used that applied these new sequencing technologies and the catalogue of genetic variation that they developed. And we've also talked about how, as these technologies continue to be applied across a wider range of projects, it's contributing to building up this library of data. And I think that finishes everything that I wanted to say, but I'm very happy to take any questions now.
Awesome, thanks so much for that, Susan. I'm aware we're sort of getting up to 5:30 because we had quite a lot of good questions sort of throughout the session, but there's a couple of questions from the Q&A I wanted to sort of pick out. One really interesting one about how the data is recorded. So someone's asked, when you sequence a genome, is every base and every chromosome recorded, and is the data kept separate from each chromosome? So is it all just put together, or is it all sort of chunked into the chromosomes that it came from?
So it's a really good question. And actually, to give you a proper and truthful answer to that would take really quite a long time. So I'm going to try and in the interests of time, keep it simple. It's also worth saying that this is something that has actually changed over time as the technologies have changed. But where to start? So I think if we stick to the kind of short read Illumina type technologies and the 1000 Genomes project used, it's a somewhat complicated process, but not that complicated. So for the short read stuff, basically what happens is that you start off with a sample of DNA, which will contain all of the chromosomes. They're all going to be in the mix there together. And what happens then is that they go through a process called library preparation. And library preparation is where you go from having your chromosomal DNA to having something that you can basically something that you can put on the sequencing machine. And what will usually happen in that, in various forms, is that the DNA will be sheared. It'll be broken up into fragments. Usually you're going to have some rough idea of what the fragment length is for these, and often it'll be maybe a certain fragment length that has been targeted. And then what also happens is that there's an additional kind of preparation step where additional bits of DNA get to the chromosomal DNA fragments. And these are called adapters, and these are able to be used in the sequence where it needs to stick in order for the sequencing reaction to happen. Then what happens is that, I think let's say maybe about 250 base pairs might be a kind of reasonable sort of length for sequencing read. So what happens is, at the end of this process, you get your little bit of DNA, which could be from any chromosome, and you've got this back, and it's got hopefully about 250 base pairs worth, say, of chromosomal sequence in the middle, plus these adapters. And what would usually happen is when the data has come off the machine, the data will come back, or should likely come back, in a format which is called FASTQ. So FASTQ is a very simple format. Basically, it's kind of like a text file. It's got a few different columns. The first one, columns, a few different rows per record. It's got about 4 different rows per record. And the two that you're probably mainly interested in are the ones which give the sequence of bases that came off. So it's just going to be things like A, C, G, and T. And then there's a row that runs in parallel to that, which records the base quality. So how confident the machine was when it made that call. And then when you've got that data back, usually what then happens is you go through a process called adapter trimming, for example, where you get rid of these sequences that were kind of tacked on at the end of your fragment of chromosomal sequence. They were basically just part of the process. So you get rid of those, and then you align the reads back to the chromosome. You align the reads back to your reference genome. So at no point are you necessarily segregating by chromosome. So you would likely have all of the chromosomes together. But yes, is a kind of complicated process that you go through. There's also all sorts of different sequencing protocols that you can use, and all sorts of different technologies that can let you look at getting bits of sequence which are maybe, are from opposite ends of a much longer fragment of DNA. And these can be helped to use to resolve structural variations. There's a whole variety of things there, but I hope that helps to answer your question.
Awesome, thank you. Yeah, it's all. Sorry, there's a whole load of questions just come in. I'm just trying to pick through them and answer maybe one or two more before we finish up. There was a really interesting one that I've now can't find. Oh, here it is. Why do we always see mutations that cause rare or harmful genetic disorders rather than seeing any positive or advantageous genetic mutations? Which I think is a really interesting one, because yeah, I think there's usually more interest in things going wrong in genetics where compared to say things that might be a benefit to humans.
Sure. So that's an interesting question, and I have to say it's not one that I have actually given a huge I mean, my guess is that often, I mean, I think in some senses we may be coming back in some respects, if we're talking about characterized variation and characterizing variation that causes disease versus, say, characterizing variation that may in some way be protective, if you like, against disease. So why do we focus on the genes or the mutations which might contribute to you having high blood pressure rather than the variations that might contribute to you having really good blood pressure? And I guess part of that is because often the motivation for doing these studies is to look at disease. It's to look at the cases where there's something that's causing someone a problem. And so it will maybe often be centered or have been funded either by people who have an interest in a specific condition. You might imagine, for example, say the British Heart Foundation, if I'm talking about blood pressure, or the Medical Research Council, or some other biomedical based focus, and they would likely have contributed a lot. There are, however, cases where you do get people who are, if you like, in inverted commas, healthy. So I mentioned before about GWAS studies, and in those, they often take a kind of, or you need to have a control set as well. So you need to have people who have the condition and people who don't have the condition. So the don't have the condition people are also there, and the sequence data is also generated. But it tends not to be the focus of the research and what people are looking for, because often what people are looking for is the thing that's causing a disease, so that you can then use that as an extra piece of information and understanding the mechanism for a disease or condition, and then figure out how you might intervene to make their lives better.
Awesome. I think the only other thing that comes to mind when I read that question is occasionally in sports, people do look into why some athletes are maybe more predisposed. So the one that comes to mind is Michael Phelps. And I think there was research that he does have some like low level genetic difference, that I think he makes less lactic acid, which basically means his muscles won't get tired as quickly. So that there is sometimes sort of advantageous mutations that are researched, but on the whole, yeah, definitely there's more emphasis on working out why a disease is caused rather than why someone's slightly better at running than the rest of us. I think looking at time, we're going to leave it there. Thank you very much to Susan for joining us tonight, and thank you for everyone for coming along to this session. I'm going to share my screen just once more with some information about our upcoming sessions. So as I said at the beginning, we've got 3 more sessions in this series, and our next one will be looking at how genetic variation is researched in the lab. So I saw a few people asking about how this sort of data was collected. If you come along next week, you'll find out a little bit more about how some of this data is sort of collected from patients. There was also a question about reading materials, and we've actually got a winner genomics textbook link in there as well. I'll pop all of these links in the chat box as well, so that you can sign up for our next week's talk and you can try and win a textbook as well. If you've got any questions that we didn't get time to answer today, do feel free to email us on the email on the screen, and that's also in the chat box. We'll try and get back to you and answer maybe a few more questions. But thank you very much everyone for coming along, and hopefully we'll see you next week.